Good morning and good afternoon. Thank you for joining the third session of our 2020 virtual user meeting. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentation. By default, all audience members are muted until the end of the presentation, at which time we would open the floor to some questions. If you have questions throughout the course of the presentation, we welcome you to add this into the chat box. It is now my pleasure to introduce Dr. Lei Wang from the Broad Institute of MIT and Harvard. I will now hand over the presentation to Lei.
Okay, so first I want to thank Nanion for inviting us and giving us this opportunity to share a tool we have developed over the past few years. So yeah, we have been using SynchroPatch system for drug discovery and also for analyzing genetic variants of ion channels. Based on our experience, we have realized usually the bottleneck of the SynchroPatch system is usually data analysis. So that's why we decided to develop this software trying to automate the data analysis. So yeah, anybody here is familiar with the SynchroPatch system. It's such a powerful system. I mean, it allows us to record from 384 cells per plate, and we can easily run 3 to 4 plates per day, getting data from over 1,000 cells. But we have realized, okay, we might actually spend days, if not weeks, analyzing the data at the beginning of our SynchroPatch experience. So at that time, we were actually heavily dependent on Excel templates to do all the analysis. And based on our experience with all of our collaborators, seems like Excel is a very common way to analyze data, but it's very time-consuming and labor-intensive. So the SynchroPatch system comes with this software, Data Control 384. So this is a good tool for exploring the data, browsing the raw data. You can quickly look at all the filters. You can set the filters to serial resistance, capacitance. There are some standard filter you can set here, which is very convenient. And you can also select the different cells, look at the raw traces. You can also select different cursors here and display the plots. So it's a great tool for browsing the data. And it can also export the data to CSV format. But based on our experience, even after export all the data from data control, we still need to spend a lot of time QC the cells and doing a lot of data aggregation, statistical tests. And that part is very time consuming. So that's what led us to develop this PatchAnalytics system. So here I will list all the reasons why we built PatchAnalytics. The first one is very obvious. Analyzing SynchroPatch data is very time consuming. So we need an automated pipeline. The second part is we also need a more comprehensive QC methods. So the patch control or data control software already comes with some standard QC methods. But based on our experience, usually we need more stringent filters based on our specific need, and those ones are not supported by default in data control. And we also need some additional interactive metrics. I'll explain this more later on in my demo. The third one is we need to analyze detailed biophysical properties, such as VHAV activation, VHAV inactivation. And getting these metrics requires curve fitting. Actually, for this one, we need to fit 2 curves. The first one is a linear regression or a modified GHK function to estimate the reversal potential. And after that, we need to fit a Boltzmann function to the conductance voltage curve to get the VHAV. So yeah, those things are not natively supported by data control. And you can make an Excel template to do that, but it still requires a lot of copy pasting and labor intensive. We also need to analyze a large number of cells and their properties across plates. So yeah, we often need to combine data from multiple plates. And doing that in Excel, it's yeah, I mean, it's possible, but you probably don't want to do that often. And yeah, so two major applications for which we utilize SynchroPatch in our experiments are, the first one is to identify the mode of action for compounds identified in the flavor assay. So this is one of our internal drug discovery project. And another one is to analyze a large set of ion channel genetic variants identified by human genetics. So I will give a brief introduction of these two projects before I give the demo. So the first project is the in-house project at the Stanley Center. The goal is to identify CV3.3 positive modulators. So what is CV3.3? It is T-type channel for calcium channel, voltage-gated calcium channel. So CACNA1I is the gene that encodes the alpha-1 subunit of CV3.3. This gene has been identified as a risk gene for schizophrenia. And schizophrenia-associated coding variants, this specific one, R1346H, in CACNA1I, has been shown to impair CV3.3 function in HEC cells. And the in vivo mouse model harboring this RH mutation exhibits schizophrenia-related phenotypes. So based on all this data, we want to identify some compounds that can facilitate the activation of CV3.3. And hopefully that can be used to treat schizophrenia. And we have identified a set of compounds that increase calcium influx through a CV3.3 channel using the flipper assay. And in this case, the SynchroPatch recording is mainly used as a secondary assay to identify the mode of action for the compounds that were identified in the flipper assays. So that's the first in-house drug discovery project. The second project I will give a demo on is to investigate the function, to do the functional evaluation of epilepsy-associated variants for SCN2A. So this project is in collaboration with L. George Group at Northwestern University. So actually all the data I will use later on in the demo are provided by Chris Thompson, who is a research assistant professor at L. George Group. So SCN2A encodes the voltage-gated NaV 1.2 channel, specifically the alpha-1 subunit. It has been shown that genes encoding voltage-gated sodium and potassium channels represent over 30% of all the reported variants in the genetic epilepsies. And variants in just 4 genes, including SCN2A, account for 40% of variant positive cases tested for epilepsy. So based on all this evidence, the goal for this project is to functionally annotate variants of unknown significance for the SCN2A gene. So yeah, after the brief introduction background about these two projects, I'm gonna give a demo of the PatchAnalytics software we have developed. So this is a cloud-based software. So it runs on a virtual machine we hosted on Google Cloud. But for the demo purpose, I'm running a local version because the local version is the most updated version. So I'm gonna log in with my password. Okay. So after logging in, you can see there are 3 panels on the left, in the center, and the right. The right hasn't shown up yet. So on the left one, it's mainly the file manipulation area. You can upload the plate files, download the plate, and run the plate, and also combine delayed plates. In the center area is what I call the QC area, and the interactive plotting area down here. I'll explain this more later. On the right side is the area that shows the detailed metrics and raw traces for selected cells. So the first thing we can do after logging in is we can upload our raw data to the cloud. So we'll click upload plate. And inside each plate, there are all the CSV files exported from the data control. And you can use export 384 to batch export all the files. And we also exported all the raw trace data from data control. So this folder includes 384 cells. That will allow us to display the raw traces in our app. And after exporting all the data in a folder, need to zip the folder. And then we need to upload the zipped folder. So I'll just do this one, demo, and open. As you can see, it will get uploaded to the cloud. So this is a local version, so it's very fast. To the cloud version, usually a plate without the raw traces only takes like 10-20 seconds to upload. If you include the raw traces, which is like 384 CSV files, huge files, it might take up to like 1 or 2 minutes. So it's still, I mean, it's slightly slower, but it's still not that slow. After uploading the plate, you can click on the plate, and then it will display all the files in the plate. So for this, I mean, we have a specific naming system for these files, because this plate is mainly for, it comes from the NAV 1.2 experiments. So we actually designed protocols only for this plate. So the plate name has to match with our protocol for it to work. And we can download the plate. I'm not gonna download it right now. We can also run plate. Let's try to run the plate. You can click on run plate. As you can see, it'll pop up a window here and ask you to select the protocol. We know this one comes from the NAV 1.2 protocol. So I'm gonna select NAV 1.2. And then all the protocol details is gonna show up here. And then the first option is ask us to select the method to estimate the reversal potential. There are two options, either the GHK or the linear regression. I'm gonna do the linear regression because it's faster, just for the demo purpose. So confirm running. And then down here, there are all the detailed QC criteria the user can set. These are the criteria. The default values are the ones we have agreed on, but we can still modify that if something, I mean, depends on the data output. So you can see we have, in addition to the standard, the field resistance, the series resistance, and capacitance, we also have the peak current QC, IV jump, leak current for pre-pulse, leak current for steady state. We also have a QC for Boltzmann slope and the reversal potential. So yeah, we'll let this one run finish. So, yeah, usually, if it's a linear regression, it should take up to 1 minute to run the protocol. If we do the GHK function, it might take longer. Naturally, I don't know, because my system crashed right before the presentation. I don't know if the server got crashed. So let me try to refresh the page, and we can give it another try. So what this protocol does is that first we'll try to run a GHK fitting or linear fitting to estimate the reversal potential for 384 IV curves. So if we choose the GHK function, it's gonna take very long. So yeah, it goes, let's see, yeah, it's running fine. And then the second step, it will convert the 384 activation IV curves and 384 inactivation IV curves to the GV curves. And the third step is to fit the Boltzmann function on the activation GV curve and inactivation GV curve to get the VHAV activation and the VHAV inactivation. And then it'll analyze some additional protocols. The final step is to filter the cells based on the QC criteria and the 2 standard deviation criteria. So it's already 99%. Let's just wait a little bit. Oh. Yeah, I think the server might have crashed right before I think it's running fine. I just give another couple seconds. Yeah, I think, you know what, I think I'm gonna restart the server. Okay, I'm gonna restart this. Okay, actually the run just finished right before I restart, because as you can notice, this area lights up. Yeah, this area just lights up. This is a QC area because after the run, some new files is gonna show up in the folder. There are some highlighted ones, the blue ones, which are the generated plots. And also it'll include the data summary. You can see there's a summary CSV and summary filtered CSV. The summary CSV is the file that include all 384 cells. And the filter ones are the only include the ones that passed the QC criteria. So now let's look at the QC area. So the green ones are the ones that passed all our QC criteria, and the red ones are the ones that failed. You might say, okay, this plate looks like the pass rate is low. Well, one major reason is because we set a very stringent QC criteria. If you simply filter the data based on individual QC criteria, which you can do by clicking on each specific one, for this instance, the capacitance, you can see the majority of the cells passed the capacitance filter. You can also look at the serial resistance. Yeah, most of them pass. Serial resistance, the majority pass. But if you look at the reversal potential filter we set, so we were able to filter more cells using this filter. We also have this peak current. Yeah, we also filtered a lot of cells here. And the Boltzmann slope, yeah, filtered a lot. Let's go back to the QCR. So as you can see, we need to use this more stringent QC criteria to make sure all the cells that pass the QC are actually good cells that's expressing the channel we expected. And we can also check this channel type. This will display the channel layout. It's color coded for the specific channels we run in each column. As you can see, the first column in the blue color is the wild type NAP 1.2. There's also some other channel variants. It's just a convenient way to remind the user about the plate layout. Let's go back to QCR. So after this step, the user might be interested in finding out, exploring the data to see to check the raw traces to see whether how come like this cell failed the QC, how come the other passed. And to do that, you can just click on any of the cells. We can just randomly click one, B6. After clicking the cell, all the detailed parameters from this cell is going to show up here. As you can see, it includes the well ID, the plate ID, the channel type for the cell, and whether it passed the QC or not, the reversal potential, serial resistance, capacitance, and the max current density, max current for the activation curve, max current for the inactivation curve. We have activation, we have inactivation, and the tau inactivation, and also the Boltzmann slope. So basically, all the parameters we care about, we can get it to display here. And it also displays the IV curves and IV traces. And for this plate, because I didn't include the raw traces, it's not going to display that. But we can actually check another plate I uploaded earlier, and we can after clicking on the cell, it can also display the raw traces to make it easy for user to identify, to confirm that the QC criteria is working as expected. And in comparison, we can try to click one that didn't pass the QC. As you can see, this one just looks weird. Let's try another one. This one, it doesn't look that bad, but it failed some of our QC. And if you want to know which QC it failed, you can just select the different filters to figure out which one. Or if you remember the criteria, you can check this detailed metrics to see why this one didn't pass the QC criteria. So yeah, QC is a very important step. So we made this interactive QC viewing window to make it easy for the user to explore all the QC filters. And now let's go back to the data folder. In the data folder, there are some pre-generated plots, and we can click on the plot. Then the plot is gonna show up down here. And this is the max current plot for different variants we run in the plates. And so the program in the background automatically does the plot. It also automatically runs the one-way ANOVA test and runs the post-hoc comparison using Dennis test. And it automatically labels if any of the groups are significantly different compared to the wild type here. In this case, there's no significance. But if we look at the VHAF activation, you can see these two groups are significantly different compared to the wild type, and it's automatically labeled in the plots. We can also look at the VHAF inactivation. Yeah, seems like these two groups are also significantly different. So yeah, everything is automatic, the plotting, the statistics, the labeling, so it just makes it convenient for the user. And in addition to the standard metrics, we also have a recovery protocol. So in this protocol, we basically give the cells like 2 pulses and then give some time to recover in between, and then look at the relationship between the recovery time and we measured the percentage of the second pulse, amplitude of the second pulse, right, relative to the first one to indicate the recovery. So the shaded area here indicate the 2 standard deviation range. So here it doesn't look like there's much difference. And we also have a protocol. This one is the frequency dependent rundown. What we did is we just give the cells many pulses. In this case, around 30 pulses with different frequency, and look at the current rundown. Here we are not seeing much difference, neither there seems to be a lot of variability. And these plots are a nice way to automatically, I mean, they can be automatically generated. And if they're good, if everything works, then we can use these plots for presentation or for publication. But sometimes we actually want to explore the plots more. For instance, in this case, you see this cell here is actually much different than the rest of the group, even after all the QC criteria. In addition, actually, there's one thing I want to emphasize. After you click on the plot, you can see in the QC area, there are some purple colored cells. And these ones are the ones that passed the initial QC criteria, but they failed the statistical outlier test, which is a 2 standard deviation range. They fall out of that range, and they are highlighted in purple because we want to differentiate these ones compared to the ones that passed the hard-coded QC criteria, so that the user will be able to make a decision themselves whether they want to include these ones or not. But even after all the filtering, multiple rounds of filtering, we might still have some cells that may give us some problems. So in the traditional static image like this is what most users usually get either from their Excel or their plotting software like Prism. They can get a plot like this easily. But then if this happens, it's very difficult to explore what happened to this cell. What's the ID of this cell? You usually have to go back to your raw data, dig through all the data, trying to find out the max current values, and then identify the ID of this cell, and then try to check all the other metrics from the same cell to see if there's anything wrong. And to make this process easier, I actually implemented this interactive plotting method here. So you can just select the different metrics to display on the plot area. In this case, we want to do the max current density on the x-axis, and the y-axis shows the channel type. And here, the same plot is going to show up compared to the pre-generated plot. We can do a comparison. The sequence, the order on the y-axis is slightly different here because this one is alphabetic order. But anyway, the data is identical. As you can see, this is the cell we saw here. And the difference between this one and the one I just showed is this one is an interactive plotting area. That means if you move your cursor to the cell, it's gonna tells you the well ID, the place, like all the basic information about this cell to make it easy for you to like troubleshoot, to identify if there's anything wrong with this cell. And you can even click on this cell, and then all the raw traces, IV traces, activation, inactivation is gonna show up on this area, in addition to all the details here. So in some cases, you might be able to quickly identify if there's anything wrong with this cell just based on these raw traces. In this case, it's difficult because the raw trace seems okay. Maybe the current was kind of big, maybe compared to other ones. But otherwise, this looks like a good recording. So we can, well, maybe some experienced person can tell if there's anything wrong. I'm not really an expert on this channel, so I can't. But to make it easier to do additional investigation, we can actually explore the data from this area further. Instead of plotting that to the channel type, we can select, for instance, capacitance. And then it'll plot the capacitance value relative to over the max current density value. And you can see this is the cell we saw before, A8. And looks like the capacitance value is fine. It's definitely on the lower end, but it's still within the normal range compared to other cells. So let's look at the max current. Of course, it's the same value, so the correlation is going to be 1. Let's try VHAV activation. Looks like the VHAV activation of this cell is much lower than actually it's lower than all the rest of the cells. So that's a flagging sign. Let's look at the VHAV inactivation. It seems like it's in a decent range. Reversal potential is definitely on the higher end of the overall range. Seal resistance 500 looks decent. Serial resistance, it's okay, 5.6. And tau inactivation is also okay. It's on the lower end, but it's not that different than the others. Boltzmann slope seems like the fitting for the Boltzmann function, the slope was much smaller than all the other values. So this just give it an easy way, another way to look at the data. So maybe you can identify some problem with this cell after viewing all the data like this way. So if, just say if, let's assume like after all the investigation, the user decided that this cell is supposed to be an outlier, it shouldn't be included in the data analysis. And you want to quickly explore what's going to happen if we just remove this cell from the data analysis. And there's one convenient way we can do it here. You can just click manual filtering mode. Then another plotting area is going to show up. And in this area, it only has all the scattered dots. And what you can do is you can use your mouse and select the area you want to plot, so that this area is going to be updated, and it only includes the data from the selected ones. And it will recalculate all the mean, standard deviation and give you the results. And you can see after the filtering, looks like this adult, this variant are almost identical compared to this one in terms of max current density. Anyway, this is just a convenient way for the user to quickly filter the data. You can even filter on individual variants and to quickly explore the data. Yeah, very flexible. You can also filter based on this scatter plot. You can just select the cells like this, and then the linear regression, the correlation, everything's gonna get updated while you are selecting the cells. Yeah, it runs very fast. Okay, that's it for the max current density for the interactive plotting. Let's go back. Oh, another thing I want to mention: if after all the investigation you feel like this cell should not be included, and I think with the manual filtering process, it's a quick, easy way to explore the data. But a better way, a more recommended way is to, after like all the investigation, we recommend the user to redesign maybe update their QC process, QC criteria based on all the investigation here. And then so that the same criteria can be applied to all the future experiments. So yeah, this plotting also helps with that purpose, because you can see here, maybe the Boltzmann slope, just for instance, maybe it should be set slightly higher. It really depends on the experiments. This is just an example. So yeah, that's it for the interactive plotting area. Let's go back here. Yeah, we can delete this plate. We don't need that one. And we have two demo plates here from the NAV 1.2 experiments. Another use case is like we usually want to combine multiple plates. And if you do that in Excel, you have to copy, paste, maybe rerun your template, but your template might not be designed to handle multiple plates, and that can be an issue. But in PatchAnalytics, you can just select the multiple plates and then click Combine Plates, confirm combining. It takes like 10 seconds to run, and let's just wait. And after combining the plate, it's gonna show a new folder is going to show up. And the name of the folder is gonna contain the name of the two original folders. Give it a little bit time. Okay, now the combination is finished. So this is a new folder that's generated. Let's click on this one. You might realize, okay, how come this area is still gray? That's because this folder contains data from two plates. That's like 700 something, 60, I forgot. Anyway, it cannot be directly displayed in one plate, but you can actually select a subplate within the combined plate. So if we click demo 1, it'll show up the QC for demo 1 plate. And demo 2 shows up the QC for demo 2. And we can look at the plots. As you can see, it contains more variants, and this one seems to be significant. Yeah, everything can be automated. Yeah, this can save the user a lot of time trying to combine the different plates. And yeah, that's all for the NAV 1.2 project. We also have some sample plates from the in-house drug discovery projects. This is a project to identify the CV3.3 activator. And we can quickly explore some of the data, but it's very similar to the NAV 1.2 project. But the difference is like we don't really have those ramp protocol or the recovery protocol or the frequency dependent rundown. We are more interested in looking at the VHAF, activation, inactivation, max current density, and the tau values. Let's look at one of the max current density. We can see our compound. So the top one is the positive control. The bottom one is the DMSO. And our compound seems to, this specific one, we hide the name of the compound. This compound seems to increase the max current density at 1 micromolar and 3 micromolar. We can look at another plate. Looks like this one only significantly increase that at 1 micromolar. And we usually need to run multiple plates for the same compound and then combine them. We can easily combine them here. and it'll give us updated data. The difference between this combination and this combination is for the nav 1.2 process, two plates we actually run different variants in two plates. So after combination, we'll have more groups. But for this project, these two plates are identical. They have exactly the same concentration, same compound. It's just like repeated experiments. So combining them allows us to have more samples within each group, and hopefully give us more confidence in the interpretation of the data. We can look at the max current density. Yeah, max current density. As we can see, these two doses are still significant. So yeah, just a good convenient way for us to combine the plates and make a better decision about our compound. Yeah, I think that's all for my demo. I'm gonna go back to my presentation. Which page? This one. Oh. So yeah, just a quick summary. Yeah, this is just a summary comparing what PatchAnalytics can provide in addition to data control 384. The first advantage is, of course, it provides automated data analysis. It takes between 1 to 6 minutes to analyze each plate. And we also provide customized QC methods. And in addition to individual traces, which is available in data control, we also provide table view of measurements derived from the traces. We also support customized data aggregation methods, and also the curve transformation, reversal potential estimation, and automated statistical tests, the one-way ANOVA and the post-hoc tests, automated data summary plots, which is interactive, and integration of QC and dynamic data summary plot. I think this one is really the highlight. It really helps the user to filter the cells to identify any issues for individual cells. And then we can combine multiple plates. Another point is like this software, because it runs on the cloud, so it's cross-platform. That means you can use your Windows or Mac to run it. It's system independent. And you can also run it on Linux. And yeah, the last one, I want to mention is like PatchAnalytics still relies on the CSV files exported using the data control or export 384. So right now we cannot directly process the raw plates. So we still need to export the data. So it mainly focuses on the post-processing of the data after you export that from data control. So here's our future plans. One thing is like we want, as we have demonstrated, the two protocols I demonstrated, they are both customized to our own needs. If we got a new collaborator and they want to use our platform, right now it's not gonna work because their like protocol tree in SynchroPatch might be different, and it might not support that. So we're working on developing a more generic protocol. So hopefully this can reach a broader user base, but there are still some challenges here because each person set up their protocols in their own specific way. It's difficult to get a generic protocol, but we think we might be able to get one with some basic function, limited function, so that at least the users can view the QC, standard QC, and take advantage of all the simple data filtering process and the interactive plotting. It might not be able to do all the detailed curve fitting because those things are like project specific. It requires a lot of customization. And yeah, we're happy to share our experience and we're happy to collaborate. But I want to emphasize that we're not a software company. We are a small team. We are mainly a science team. We have a very small software team. Actually, I'm only like half computational. My main focus is still science. I'm a scientist more than a software engineer, but we're still happy to help. So yeah, right now we have limited bandwidth. If you wanna use our platform right now, we would love to help, but the current setup is not gonna work. We might be able to help you set a protocol, but that may take a lot of time. So right now we have limited bandwidth. We're not optimized to do a broad-based software development. But I still encourage everybody to reach out to us. If many people are asking us for, if we get a lot of interest from a lot of people, we might be able to, put more resources into this software development. But the current way it is, it's mainly used for our own needs. Yeah, I want to acknowledge everybody involved in this process. We cannot develop this software without all the support. First person I want to mention is David Zhou, who is a software engineer in our group. He set up all the cloud environment, configured all the virtual machines. He also laid out the foundation for the framework. David Bias, he's our electrophysiology project. And yeah, I just simply translated his Excel template to a Python language to make it work in our software. Also want to acknowledge our collaborators at Northwestern University, George, Chris, and Carlos. I want to mention Chris because for all the Nav 1.2 data analysis, he provided all the Excel template. And I went through the template and translated all the template into Python. So it's not going to work without their help. I also want to thank Nanion Technologies. They are the group that helps maintaining our synchro patch system and making sure it works properly. And I really appreciate all the customer services. Their Tim and Leo, they're usually very quick in responding our emails. All the emails get responded within 2 to 3 hours. So really thankful for that. And the last thing I want to mention is our funding resource. So some of the funding comes from Stanley Center, the internal funding. Some of that comes from the U54. The principal investigator for the U54 is L. George at Northwestern. And yeah, that's all of my presentation. And I'm ready for any questions.
Thanks for the presentation, Lee. I'll go ahead and moderate now. I know we have a couple questions received, so let me just get right into it.
Okay, should I just stop sharing now?
Yeah, you can leave it on this slide. It's fine, or just you can stop sharing. It's okay. Either or works.
Okay, I'll just leave it this way.
Okay, so in terms of the what you presented to us, how long did it take to develop this?
Oh, yeah. So total, it's actually several years of effort. So I joined the group in January this year, but before I joined, our group has already contracted a software company, and they developed a prototype, a simple version, a local version, which runs okay, but it's not as interactive. And we have used that software to do our analysis, but it's not flexible. So after I joined the group in January, we decided to make the software more flexible, more interactive. So I have spent a lot of time in the past, like 9 months, trying to move it to the cloud, adding all the interactive functions, all the QC plots. So yeah, I would say in total, I don't know which year they started, but it's several years of effort from multiple groups, companies.
Yeah, and then as a follow-up question, is this available to other SynchroPatch users? And then additionally, when you mentioned the combination of plates, how many plates can you combine in total?
Oh, right now, I haven't tested the limit, but the code is written in a way that it can handle unlimited plates. But I'm sure it depends on our virtual machine resource. If the plate number gets too high, we might not have enough memory. Right now, we usually just combine 3. So, but if we have the need to do many plates, we can upgrade. We can check the system, the hardware, make sure all the resources is there. But the code is written to handle as many as possible.
Understood. And then I think David is eager to ask you a question, so I'll pass over the mic to him.
Okay.
Hello.
Hello.
David Cahoon. I have a question about how you do the averaging. If you're fitting say, an IV curve,
Mhm.
and you're averaging it from dozens of cells,
Mhm.
do you fit each cell individually and average the parameters, or do you average the data and fit once?
So for the IV file and then the average, let me try to recall. Well, right now we haven't averaged the IV curve. Yeah, we mainly get all the metrics from the curve. Like right now, let me go back to this.
Well, any curve, really.
Yeah, so for the max current density, yes, we get all the cells for like 384 cells, and then we filtered the ones that failed the QC. When they do a second round filtering of the 2 standard deviation. So the first QC process, like this QC, it's on a cellular basis. So if the cell failed the QC, it's gonna be removed from all the analysis. And then the second round statistical 2 standard deviation test, it's a metric specific criteria. So for example, for this max current density, if some cells failed the 2 standard deviation test for this metric, it's only removed for this metric. It's not removed for other metrics. And then we group based on the either cell type, channel type, or compound type.
If I could jump in, I think what David is asking is whether or not we average for each cell and then for the entire plate. And then the answer is yes, we calculate for each cell for the GV curve, and then we calculate the difference across different cells within the same plate or across the plate.
Oh, so that would avoid the problem of averaging the slopes,
Right.
because it's a sort of well-known thing that if you have curves with identical slopes that are shifted relative to one another, if you average the data, you get a slope which is shallower than any of the observed ones. But you do it in a way that avoids that.
That's correct.
Good.
To be honest, and so this is Lei's effort relatively recent, and we haven't tested in many of our existing data, but so far there aren't that many variants that influence the slope, that the few that we've seen. But yes, that's absolutely correct.
Thank you.
Thank you, David.
I think I'm on mute, sorry. Another question that came in regarding the compounds that were tested on CAV 3.3. Did you test any compounds that were identified by Flipper which did not activate or block the current as you would have expected when tested on the SynchroPatch, i.e. did you have any false positives from the Flipper screen?
Yes, we actually do have some compounds that were positive in Flipper, but it didn't increase the current density in the CV3 in the SynchroPatch system. So I think Flipper assay is a calcium signal, based assay. So there are some caveats. So that's why it's a great way to do an initial screening. But for more detailed like biophysical properties, maybe SynchroPatch can provide more detailed data. So they don't always align with each other. But in that case, I think SynchroPatch is, the data from SynchroPatch is more, in a certain way, more convincing.
Okay, perfect. Well, Lee, thanks again for going through the presentation and doing the live demo for us and running through the software. I'll make sure that I'll go ahead and tag all the information in the backlinks in the description of this on-demand copy. And then I'll make sure that there's any additional links to your group. So if there's any questions externally or collaboration needs, they can get in touch with you guys directly as well.
All right, thank you.
Thanks, Lee. Thanks, Jen. Have a good day now. Take care.
Thank you. Thanks.
Thank you. Bye.